I have a very wonderful job to do, which is to award another one of the BSDB medals. This one is the Beddington Medal, which is awarded for the best PhD thesis in a year, in this case 2016. The nominations for next year will open soon, so if you have an excellent PhD student or know of an excellent PhD student who has submitted or will submit a thesis in developmental biology this year, then please do nominate them. The winner, though, for this time round, I'm delighted to announce, is ErikClark, who has finished his PhD recently in Mike Akam's lab. And we were extremely impressed by the amazing work he'd done that he will be talking about now, which illustrates, I think, a really interesting issue in developmental biology, which is the relationship between space and time, and how one can accidentally confuse those things if one's not careful. And then on top of that, something close to my heart, the extraordinary power of computational modeling in allowing one to disentangle those things in one's mind, where sometimes that relationship is not intuitive. It can become intuitive through the process of building models. So I hope you'll join me in congratulating Erik very much on his amazing achievements and in coming to present his talk.
So good evening. I'm really pleased to have been awarded this medal. It's a real honor and a privilege. I'm also immensely relieved that the 3 trains, 2 planes, 2 buses, and a taxi that took me from San Diego got me here in time and only slightly jet-lagged. So I'd like to thank the organizers. I'd also like to give a big thanks to my PhD supervisor, Michael Akam, who not only gave me great encouragement and support and also academic freedom, but also permitted me to reintroduce Drosophila to his lab after a lengthy absence. So Drosophila are the subject of my talk today, and some of you might wonder whether there is anything new to say about Drosophila segment patterning, but I hope to show you that there is. Okay, so let's start with some arthropod embryos, and you can see their sort of conserved organization into a segmental structure. The segment boundaries are established early in development and marked by stripes of engrailed. And this conserved modular body structure is thought to have facilitated the evolution of the huge variety of adult morphological forms that we see in the arthropod phylum. So this idea is basically like the top half of the so-called developmental I'll set my timer. Developmental hourglass, in which we start with a phylotypic stage, and we can get from there to many different adult forms adapted to different ecological niches. The flip side of this idea is that we can get to the phylotypic stage from many different starting points, and the early embryonic stages are also adapted to different ecological niches and reproductive strategies. So we see a huge diversity of early embryonic forms within the insects. We've got this huge array of shapes and sizes of insect eggs, and I'd like to also point out the important distinction between long germ and short germ modes of development. So the germ refers to the embryo proper, which is the dark gray regions you see in these pictures, and in some insects like Drosophila, the embryo fills the entire length of the egg, and they're known as long germ. Whereas in other insects, like Tribolium, the embryo only fills part of the egg, and they're known as short or intermediate germ. So long germ and short germ development are associated with different modes of segment patterning. Long germ insects like Drosophila, you have all 14 engrailed stripes emerging simultaneously within the blastoderm before germ-band elongation, while in short germ insects like Tribolium, you have those same engrailed stripes emerging sequentially at the posterior in concert with axial elongation. Short germ segmentation is the ancestral state, but long germ segmentation has evolved independently several times, raising the question of what exactly is the relationship between long and short germ modes of patterning, and what underlies the apparent ease of getting from one to the other. We, of course, know a huge amount about the molecular mechanisms underlying long germ segmentation, thanks to the famous Drosophila segmentation cascade, we go from the, in which you've got this hierarchical cascade of transcription factors which iteratively subdivide the embryo into finer and finer units of space, going from the broad maternal gradients through to the zygotic gap genes, through to the emergence of periodicity with the pairal genes expressed every other segment, and finally the establishment of discrete cell states with the segment polarity genes. We know much less about the underpinnings of short jump segmentation, but a couple of things are pretty clear. Firstly, the early players in this cascade have a much smaller role in the entire process, and instead, orthologs of the pairal genes have this really interesting expression where they actually oscillate in the growth zone over the course of germ band elongation, and you also have stripes of pairal gene expression emerging anterior to the growth zone. And although we don't know, exactly the gene networks involved, it seems pretty clear that what we have here is some kind of clock and wavefront system. So just so we're on the same page, I'll show you a really simple Boolean model of a clock and wavefront system. We're going to have two genes, clock and wavefront. Clock is going to auto-repress itself if wavefront is on, and it's going to, wavefront is off, and it's going to auto-activate itself if wavefront is on. And wavefront, in turn, is going to turn on progressively across this idealized tissue of 20 cells. One more thing to remember, I've added some time delays to the system. So if you see a nice kind of solid rectangle like that, that means protein. And if you see a hashed rectangle, that means transcript. So if I press play, okay, so you see that the clock gene is oscillating continuously between transcript and protein. And then as wavefront turns on across the tissue, those oscillations get frozen into segmental stripes, into periodic stable stripes. And so this nice periodic output gives us stripes, but it doesn't give us segments. Segments, at least in Drosophila, are a whole lot more complicated than simple stripes. And to illustrate that, let's look at exactly what the pairal genes have to do. So here we have a triple antibody stain of 3 pairal genes, Eve, Futz, and Runt, in a cellularization stage Drosophila embryo. And you can see this kind of nice rainbow repeat pattern along the axis, which repeats every sort of 7 to 8 nuclei. This pattern arises because each of the 7 pairal genes are expressed with a slightly different relative phasing along the AP axis. The positional information contained in this repeat is transduced by about 30 minutes later at gastrulation into this nice digital segmental pattern in which each of the nuclei in the trunk express one and only one of these 3 genes engrailed, odd, and sloppy paired. And this transduction process is entirely done by transcriptional regulation, so we've got no cell divisions, no cell rearrangements, and no cell signaling going on during this time. So one thing to remember, right, so we're going from this repeat here through to this repeat here, and this is the one thing I want you to remember. We've got this nice output pattern that goes engrailed, odd, sloppy paired, engrailed, odd, sloppy paired. The basic idea underlying how this is achieved is pretty old. It's actually old, a little bit older than me. And basically, the idea is that you have different pairal genes expressed with different relative phasing along the AP axis, which gives rise to different combinations of transcription factors in different nuclei along the AP pattern repeat, which sets up a kind of combinatorial code which is sufficient to establish discrete segment polarity fates. Okay, so fairly complicated already, and I'm going to add an extra complication, which is the temporal dimension. So if we look at sort of early cellularization, when the pairal genes have their periodic pattern established by the gap genes, we see the pattern is kind of rough and ready, and it's only later, at sort of mid to late cellularization, that we get this nice regular pattern of 7 stripes of each gene emerging. And then at gastrulation, even more stuff happens, and we have several of the pairal genes dynamically transitioning from double segmental patterns of 7 stripes to single segmental patterns of 14 stripes. And this entire process is mediated by pairal gene cross-regulation. So again, the idea is fairly old. We have some network of genetic interactions between the pairal genes, which act to establish different expression states in different nuclei. And over time, this effectively amplifies positional information inherited from the gap genes into a very nice, sharp pairal pattern. However, you might have realized that these 2 ideas, these 2 jobs that the pairal genes have got to do, are sort of in conflict with each other, right? So in order to transfer information to the pairal genes, we need to have a lot of positional information in this pattern, so we need a lot of overlaps. However, in the act of organizing their own pattern and cross-regulating each other, the pairal genes will tend to erase those overlaps. So it's not the 80s anymore, and we actually know a whole lot more about the pairal gene network, but I'm not sure how much more we actually really understand. So this is the last paper on the pairal gene network before I think people got a bit fed up and moved on to other things. But we basically have this whole mess of inferred genetic interactions, but no real systems-level understanding of how patterning is occurring. So we come to these two interlinked questions. We've got the kind of nerdy developmental question of how we get from this state to this state over the course of 1 hour, and that's what I spent my PhD on. And then we have the broader context of how did this whole scenario we see in Drosophila emerge over millions of years from an ancestral short-germ state. And I mean, I just said that I spent my PhD focusing on this question, but it's biology and you can't escape evolution. And I think the things I found out in the course of this study actually shed light on the evolution question, which I found quite exciting and cool. So now I'm going to basically summarize a couple of years of my life with one slide. I spent a long time doing double fluorescent in situs of all pairwise combinations of pairal genes in wild-type and mutant backgrounds, and cross-referencing these observations with all the experiments in about 150 papers on pairal genes from the literature, and synthesizing this to try and clarify how the pairal genes regulate each other. And the important conclusion I had from this process was that the spatial information inherent in the pairal genes themselves is not sufficient to explain the spatiotemporal patterns of the pairal genes over time. And instead, we need to have additional temporal inputs from a number of broadly expressed transcription factors which turn on and off over the course of segmentation, which actually have for a long time been recognized to play important roles in the segmentation process. Because they're obviously, if you don't have them, things go wrong. So I'm going to briefly go through the roles I discovered for each of these genes in the pairal network, and then I'm going to bring everything together into a model. So first we have caudal, which is the Drosophila ortholog of CDX, and it plays important roles early in segmentation, activating gap genes and so on. But the thing I got interested in was actually the pattern with which it retracts from the germ band during the second half of cellularization. So it disappears ventrally before dorsally and also anteriorly before posteriorly. And I realized that this correlated really nicely with the pattern with which the paired stripes emerge. So, and then later verified that indeed caudal turning off is controlling when paired turns on. And this was quite nice because it explains this old experimental finding where if you misexpress caudal during the second half of cellularization, which is exactly when paired comes on, you end up with this cuticle phenotype that looks very much like a paired mutant. The second gene was dikete, which is the Drosophila ortholog of SOX2, which is also around during the earlier parts of the pairal process. And the mutant phenotype of dikete is very interesting because you get segment patterning defects, but these are actually very variable in severity. And what I found out was that dikete is required for certain cross-regulatory interactions between the pairal genes, which aren't strictly necessary for segment patterning per se, but actually increase the robustness of the patterning process. So if we look at this wild-type embryo, we see these nicely abutting stripes of odd and runt, which arise partly because the odd skip protein represses runt. However, in dikete mutants, we see extensive overlaps between odd and runt because odd fails to repress runt. And this leads to this kind of irregular pattern of runt stripes, which down the line will eventually lead to patterning defects. The final gene, which is my favorite one, is odd-paired, which is the Drosophila ortholog of the Zik genes. And odd-paired was identified through the initial segmentation screens as a pairal gene, and so about 15 years afterwards, it was assumed that it would be expressed just like the other pairal genes in stripes. But when it was finally cloned, it was realized it was aperiodically expressed and was sort of promptly discarded for future study as being boring and uninteresting. However, I realized that odd-paired, the time that odd-paired protein is turning on in the embryo is actually a really interesting time. It's exactly when all the pairal genes start transitioning from these patterns of double segment periodicity through to these patterns of single segment periodicity. And it's also the same time that all of the segment polarity genes start turning on. And indeed, it had already been noticed that in odd-paired mutant embryos, the turning on of the segment polarity genes was delayed. So I figured maybe odd-paired turning on was some sort of temporal signal which not only would activate the segment polarity genes, but would actually change how the pairal genes regulated each other and precipitate these expression pattern changes that we see in the embryo. So to test this, I looked at odd-paired mutants. And here we see the wild-type expression at cellularization and gastrulation. And as expected, in the mutant, everything looks totally fine at cellularization. But once we get to gastrulation, things start looking very abnormal. And in particular, all the pairal genes fail to go through this all-important transition from double segmental to single segment periodicity. So bringing all this together into a model, I've spared you the details of how exactly I arrived at these topologies, but we basically have two networks, one operating early, so during cellularization, and one operating late during gastrulation. The early network is taking positional information from the gap genes and feeding them through these key primary pairal genes, even hairy, through to the rest of the pairal gene system. At gastrulation, we switch to this late network, which has a lot of mutual repression, and is acting to really lock in discrete cell fates. The missing ingredient are those temporal signals. So we've got caudal there early present early, and it's acting to keep off the secondary pairal genes and basically allowing the primary pairal genes to get themselves organized before paired and sloppy paired turn on. We then have odd paired coming on late and mediating this transition from the early network to the late network. And for simplicity, poor old Dikete isn't in this model. But everything's coded up now into a Boolean network. And I should just say, so I'm not trying to fit the model to the data here. I'm trying to just see what the network wants to do. And so almost every parameter value in this simulation is set to the same arbitrary number. So my initial conditions, I've got some temporal information from, okay, I should tell you now, for paper-related reasons, wherever it says X, that means caudal. But remember, X is caudal. So caudal is going to turn off after a certain amount of time, and odd paired is going to turn on shortly afterwards. We then have some spatial inputs into the system from some abstracted gap inputs, which represent the repressive inputs to the pairal genes from the gap genes. So G1 is going to repress hairy, and G2 is going to repress Eve. So if I press play, we see that the pairal genes sort of resolve, and then the network's going to change, and everything will stabilize, and it's completely rubbish. So I told you we wanted that output pattern engrailed odd sloppy paired, engrailed odd sloppy paired, and we don't even have any engrailed expression at all. So something's wrong with this model, and one particularly obvious thing, which is completely wrong, is this assumption that these gap inputs are going to be static over time. Whereas, in fact, if we measure gap gene expression in the embryo, we see that the gap gene expression domains dynamically shift from posterior to anterior over the course of cellularization. And this is because the gap gene network is organized so as to drive this temporal sequence of gap gene expression from more anterior genes to more posterior genes over time. And so each nucleus along the axis is going to drive a partial sort of part of this sequence in the limited time it has during cellularization. So now let's put this assumption into the model. Everything else is going to be exactly the same, but now I've changed these gap gene inputs into sort of little oscillators which are going to drive kinematic waves across the tissue. So let's see what it does. So now the pairal genes kind of organize themselves, and then the network is going to switch, and look at that. So the output we get engrailed odd sloppy paired, engrailed odd sloppy paired. I can play that again for you. Things get organized, and there we go. Everything's stable with the correct output pattern. And not only is the output pattern correct, but if you look at what the expression of the pairal genes over time in the simulation, you can then compare that to real expression in the embryo, and you've got to take my word for this, or you can check my preprint because it's a pretty small figure. But it matches really nicely what the pairal genes are actually doing in the embryo over time. And I can also do things like take one of the pairal genes out of my simulation, and it will recapitulate really nicely what the mutants do as well. So it seems like dynamic gap inputs are really important for the parallel gene network to execute its function properly. And I'm going to try and explain very briefly why that's the case. So here we're looking at what's going on at the posterior boundaries of the Eve stripes. And here are the relevant genetic interactions from the early network. And you can see that if we let this go to steady state, there are only two possible expression outputs. We can have even runt co-expressed, which is eventually going to give rise to the sloppy paired output state, or we can have odd and futs, which is also going to give rise to sloppy paired. So we get a really sort of terrible output pattern there. However, if Eve turns off in a given cell, it's going to take some time for odd skipped to be derepressed and then for runt to be repressed in turn. So if this Eve stripe is moving across the tissue, then at the back of the Eve stripe, we're going to see this temporal process kind of smeared out over space. And that means that we get these transient states of runt alone, which actually gives you engrailed, and runt overlapping with futs and odd, which actually gives you odd skipped. So the analogy I like to use for this patterning mechanism is imagine that the Eve stripe is a ship, and here we see the ship is static, it's got the anchor down, and everything outside the ship is flat ocean stable state. However, if the ship is moving, we get something else, which is a wake. So that's where the system has been stirred up by having the ship go through it, having Eve turned on, and is in the process of going back to stable state, but it's going to take some time for that to happen. And we get this nice alignment of different states along the axis, which is a really good way to pattern a tissue. It also resolves that paradox I showed you earlier, because now the whole process of the parallel genes organizing their own expression, the cross-regulation, actually creates overlaps. Okay, so in the final couple of minutes of my talk, I'm going to go back to that evolutionary question which I raised at the beginning. So, and the great thing about models is that you can kind of play around with them and change things and see what happens, and you can explore evolutionary possibilities. So we know that the gap inputs are a fairly new recruit into the parallel gene network, into the whole process of segment patterning that we see in Drosophila. So how about we just take them out and replace them by some fairly ad hoc extra cross-regulatory interactions between the parallel genes, which will actually maintain the spatial patterning of these genes even hairy. So now everything else in the network is the same. Caudal or X is going to turn off, odd paired is going to turn on, but the only spatial input we're putting in is a kind of phase gradient of this little oscillation of hairy that we have here. So now the system's going to take a little bit longer than normal to get itself organized, but once it does, everything's going to happen just the same as before when we had the gap inputs in the system. So that's telling us that, because all our spatial information is just coming from that phase gradient, that sort of temporal pattern of hairy, this entire spatial pattern we get at the end is emerging from the intrinsic temporal dynamics of the parallel gene network. We can then go one step further, because this kind of initial pattern of hairy, is fairly sort of ad hoc input into the system. We can take that out and instead replace that spatial input with a decay gradient of caudal, which is going to, so caudal is going to turn off across the tissue in a wave, and then odd paired is going to turn on. Everything else is exactly the same. So now we see that the tissue at first starts oscillating through parallel gene expression, but then as this temporal signals move across the tissue, we freeze those oscillations into that complex segmental pattern that we saw before. I can show you that again, maybe. Let's try it. Okay, so oscillations, so we got a clock, and now we've got a wavefront. So the same network that was driving Drosophila simultaneous patterning is also capable of driving sequential clock and wavefront type segmentation. And we end up at that same conserved output state that we see across all insects. So we come to the hypothesis that actually, if that same network is capable of driving both modes of segmentation, maybe the network is fairly conserved between long-germ and short-germ insects, but that the temporal sequence that we see in Drosophila is translated into a sort of spatial process in short-germ insects. And actually, if you go and compare parallel gene expression between short-germ and long-germ arthropods, this hypothesis seems like it could actually be correct. But we still need something which would make long-germ insects long-germ and short-germ insects short-germ. And the obvious candidate is our temporal signals. We know they're acting as timers in Drosophila, but in order to convert the Drosophila network into a clock and wavefront system, they would have to act as wavefronts. So to test this, I got help from Andrew Peel, who has a tribolium lab at the University of Leeds, and he looked at the expression of the orthologs of these genes. And in the beetle, we see that caudal is indeed expressed as a very nice wavefront, which retracts along the germ band over time. Dikeet 2, this posterior domain, is always associated with the growth zone. And then the posterior boundary of odd-paired expression is also moving down the germ band like a wavefront. So actually, all of our cells in the tribolium embryo are experiencing this same temporal sequence of caudal Dikeet odd-paired. It's just that the anterior ones go through it much quicker than the posterior ones. And this is suggesting that actually we could get most of the way from short-germ to long-germ segmentation simply by changing the spatial temporal expression of these three key temporal inputs. So with that, I'd like to thank Michael again and thank Andrew for the tribolium work and thank all the other people who have given me advice, support, and inspiration over the years. And I'd like to thank you very much for listening.
Beautiful. Does anybody have any questions?
Beautiful. Really beautiful, and the network conservation is a great idea. Is it enough to add growth, progressive growth, to convert long to intermediate or short-term development?
So I think from my simulations, there was no growth in them. Actually, growth is a good way of producing a gradient or a wavefront which retracts over time. Because if you've got something associated with the growth zone and that's moving over time, that will create that process. But you could also think of other ways in which you could create a wavefront which would move across the tissue. But the kind of the transcriptional change I made in terms of my initial conditions wouldn't actually be sufficient to create a robust short-germ segmentation process, because actually in short-germ embryos, the cells are also going to be moving around a bit. So you'd need something additional on top of that, some kind of cell-cell communication to keep all those transcriptional patterns synchronized with each other.
One more over there.
What turns on odd-paired?
Okay, so in Drosophila, odd-paired seems to be dependent on nuclear cytoplasmic ratio, and actually the transcripts are appearing pretty early in cycle 14. However, while the parallel genes are all fairly short, they're sort of like 3 kb long transcription units, the transcription unit for odd-paired is like 17 kb, something like that. It's got a big intron in the middle. And so, I mean, we're making an antibody at the moment to try and check exactly when the protein is there and check it and cross-reference it against the original papers where we tried that antibody but didn't work on our hands to see when exactly the protein is there. But in the original papers, it's reported to be, only appearing end of cellularization. In short-germ insects, it obviously can't be that sort of ad hoc Drosophila mode of timing, and I think it's more likely that caudal Dikeet and odd-paired, or indeed their vertebrate orthologs CDX, SOX2, and ZIC, may be involved in some kind of genetic cascade which is involved in the whole process of going from stem cells at the posterior of the embryo through to differentiated cells more anteriorly, which has then been recruited into the segment patterning process in arthropods.
Thank you. Okay, I think possibly in the interest of time we should move on, but I would like very much for us all to congratulate Erik one more time on the beautiful work.